Doing this, I'm going to introduce our first speaker, Yul Belize from University of Michigan. And I have known Yul for many, many years. I've probably learned a lot about big data and digital pathology and algorithms from Yul. And many, many conversations we've had at several pathology informatics meetings and AP III meetings and others. So I'm very grateful to him for that. And I'm very grateful that he was able to accept this invitation at a very short notice. Yul is a board certified pathologist and a clinical informaticist at the at the University of North, University of Michigan. He is the medical director of the Division of Pathology Informatics and has a long-standing interest, has been on the ground working with several vendors and actually helped them build products uh, in digital pathology. He is an expert in computational pathology, data analytics, pathology operations. And today he is going to talk to us about supporting the NIDDK Kidney Precision Medicine Project and standing up the U of M program of Pathology AI and Data Visualization Center and the core lab. And they have a very, very robust program in pathology informatics, including one of the first pathology informatics fellowship. So between Pittsburgh and Michigan, if I look at all the trainees that have come out of these two places, you're going to hear from Mike Besich also later on. Many of the trainees in pathology informatics have their ties to these two institutes. So I'm very pleased and honored to introduce Dr. Belize to come and give his keynote speech. And are you able to share your screen now?
I believe so. Let's see if this works. Looks like it's working.
Excellent.
So thank you very much, Anil, for that kind introduction. Hopefully this will work. Let's see if it's starting up. It's starting up, good. In the next few minutes, what I hope to share with you is an opportunity which came from NIDDK to create a consortial effort for exploring patients with both acute kidney injury and chronic kidney disease. This is known as the Kidney Precision Medicine Project. And it happened about 5 years ago, about the same time we had our initial thoughts of standing up a core AI and machine learning lab in support of digital pathology. So it was really a perfect storm to do both of those things. And this story is by no means complete, but it's certainly well underway. And with the recent arrival of my colleague, who is now here at Michigan, Dr. Liran Pantanowicz, the pace at which we're implementing both clinically deployed machine learning, not just AP, but also AP and CP intersections. So many opportunities in lab medicine as well. But the clinical opportunities for clinical service with these tools in tandem with discovery creates really a perfect storm. So that being said, just I guess as a reality check, Anil, is the audio working? Is everything
The audio
okay?
is working, but you need to bring up your presentation mode of the PowerPoint. We can still see all your emails and everything.
Oh, let's see,
You can share, I think you just have to share the right screen.
Okay, I'm sorry, I'm glad I checked. Let me see which screen I'm sharing. I thought I was, oh, I see, I'm sharing too. I need to, let me fix that. I'm glad I asked. I thought this was
I was going to interrupt you after this first.
Okay, I see what's going on. Let me stop this for a second. I thought I had the right one, but apparently not.
Yeah, Yul, when you use the multi-screen, you probably have a multiple monitor. You got to watch the green highlight box on Zoom so it grabs the right screen.
Yeah, I see what happened. Okay. That's exactly what happened. Actually, I rehearsed this multiple times and it was the right monitor. I didn't expect Zoom to change it. So we'll try it again. screen too. I think that's right. Okay. I'm sorry about that. Let's try again.
So we can see the desktop, but not the present. Okay, now it's good. Yep.
Okay, good. I'm really glad I asked. Okay. So as a backdrop, the Kidney Precision Medicine Project has multiple goals. It's unusual in that it's not merely a discovery effort, but it has research subjects who are at the same time patients. So the idea is to identify opportunities, therapeutic targets, and metabolic pathways, as well as new cell types in patients so that the participants can actually receive direct benefit of the study itself. And towards that goal, this is really one of the first, if not the first study, at least I'm told, in NIH's and NIDDK's history, where research biopsies are being performed as part of the study, both for management of the patient as well as for discovery. So one of the goals of the Kidney Precision Medicine Project, besides the discovery, is generating a multi-axial atlas where for the first time, digital pathology itself becomes an integral portion of the data that's gathered. And not just for the purposes of having archival whole site images for inspection, but as part of an overall computational portfolio. So the images are intended to serve as one of the entry points for the multi-axial database that will also include clinical outcomes measures, long-term clinical abstractions from the chart, as well as molecular and omics type data. So the idea would be, starting with an image, which is, I guess, unusual for a database interrogation model, the image would be the starting point, which then could branch out to the various other types of curated data in the data lake and knowledge environment, including clinical context, morphologic content itself, and molecular and single cell data, so that the search predicate, for example, if it were like a disease glomerulus, could then map to a plurality of similar cases based on the morphologic equivalence, and then could map then to additional classes of data, for example, various partnering tissue interrogation sites have advanced imaging modalities, which are also being contributed, various types of immunofluorescence, for example, multi-axial, multi-channel immunofluorescence, as well as the omics data and the TSNI data, and then finally outcomes measures to generate a holistic and integrated view of the patient, which obviously would provide tremendous opportunities for incremental discovery. And in terms of the type of data that's already being gathered, multiple tissue interrogation sites are starting to create reference atlases of healthy and diseased patients. And this is allowing us now to create computational pipelines that will further classify the surface area, the images, and then where in some cases we have, for example, either laser capture microdissection or matrix acquired single cell and omics data to combine image data with proteomics and transcriptomic data that's spatially arranged to generate even more precise classifications of the histology itself as well as the disease processes. And so the metabolomics, proteomics, and laser capture microdissection are all part of this study. And this allows then for various opportunities in stratification of patient populations, of cell populations within individual biopsies, and ultimately classification of disease based on the type of cells present, as well as the clinical trajectory. So basically a holistic approach for interrogation of data, and then walking across the modalities between the morphology and the omics and other types of high dimensional, high throughput data. And this project, the NIDDK's project, really became a motivation for at the same time building our capabilities within pathology in the form of a digital visualization, data visualization core lab, which has multiple types of AI. And that was based on a simple foundational model which starts at one of the platforms really is recognizing that whole slide imaging now is mature. So incrementally getting as much of our department up to speed. We're not nearly at the level of Dr. Parwani's department yet, but that's aspirational for us. And in fact, we've acquired a number of scanners, excuse me, and we're in the process of activating whole slide digital primary workflow, one service at a time. So I would say probably within 1 to 2 years, we will be largely well on our way in that journey, using that clinical operational experience to inform the opportunities for layering on top of that the other things which you can do. Things that are obvious, low-hanging fruit to implement once you have digital workflow is autonomous quality detection systems. Actually, following me this afternoon will be a presentation. I'm sure he might include parts of it from Dr. Anat Matabushi at Case Western, who's recently just published on HistoQC, which is a very elegant pipeline for, in fact, carrying out this exact type of quality detection so that the images that are generated in a high throughput pipeline can be assured to be of a suitably acceptable minimum level of quality. Then identification of single features, which then, of course, can be constitutive to finding multiple features and multivariate feature analysis. This has been one of the primary foci of our discovery efforts in terms of machine learning. And I'm pleased to announce that the tool we've been building now in multiple generations for well over a decade was originally published as spatially invariant vector quantization, SIVQ, is now fully grown up as an application suite known as Viper Studio for validated image pattern recognition. And that tool is now finally available for download, freely available from our group. And anybody who's interested in using that tool should reach out to me after this presentation and we'll make it available to you. We're already in the process of getting it available to a number of groups, including Anil's group, for example. And then finally, what we envision, and this is really more of a long-term aspirational goal, turnkey systems which provide diagnostic recommendations as well as discovery. And I mention this with the very necessary qualification that I don't see, at least for the foreseeable future, these tools replacing pathologists, because there's still an art in the practice of pathology, and that applies also to lab medicine, for recognizing the clinical context and the morphologic context, and recognizing that these algorithms, if they're run completely autonomously, can operate in an extrapolatory fashion and yield wildly divergent recommendations from the reality, so that, you know, there still needs to be an adult in the room. But that's not to say you still can't use these tools to significantly automate the workflow so that the pathologist, for example, can operate at their highest credentialed level of looking at features that have been pre-screened so that their overall operational task is that of directed review as opposed to screening, which is both time-consuming and fatiguing. So as some examples of things we've built which supported the Kidney Precision Medicine Project, and a number of groups have done this, so I won't belabor the issue, but we actually built a very high performance viewer that allows for real-time viewing over the web. It's actually more fluid motion than you're seeing in this recorded video. And that's actually currently active for the participant viewer in the Kidney Precision Medicine Project website. But in addition, that allowed us to build a number of both thin and thick client applications that have very fast image retrieval, which then supports our computational pipelines. So maybe enough about this image. And you can see this viewer in work in action. It's publicly available at mydata.kpmp.org. Additional studies right now that we're leveraging is a top-level atlas that takes all the information that's been acquired from the multiple participatory tissue interrogation sites and puts them into one overall repository that allows for visualization analytics that operates across multiple length scales. And this has proven to be challenging. The future directions of this, of course, are creating a macro language and a repository so individual pathologists or investigators can stand up their own computational pipelines making use of the information that's available at the multiple length scales. So what this involves really schematically is single cell omics data, 3D imaging FISH, Codex data, which is, for example, immunofluorescent stains of 50 to 100 or even more, 200 channels of the exact same sections so that image information for different markers is spatially exactly co-located, regional omics, and then finally metabolomics, all blending it with a digital pathology repository. And this type of data acquisition is well underway. Tara Kashkar's group, for example, has a working crosswalk between the RNA-seq and imaging data, which correlates very, very tightly. And that allows us then to match to the immunofluorescence acquired large field images and showing the spatial organization, for example, of healthy and diseased kidneys, a type of structure we're seeing really for the very first time. And this is allowing us to generate a number of hypotheses as to the evolution of acute kidney injury into chronic kidney disease. A very exciting period. Another type of task, although mundane, can be extremely tedious, is simple annotation and ground truth maps. We've really, I think, extended the notion of this, and this will be the latter portion of my presentation, really. It will be interactive, showing what Viper Studio can do right now. And we're adding features to it because the back end of it now is a full-featured machine learning classification engine. But in order to generate precise ground truth maps, the old reality was that it was a very time-consuming process that is or can be at least subject matter expert dependent, where one individual might make more precise annotations than another individual, and that individual might be inconsistent over space and over time. And of course, as we know from machine learning, inconsistency in the ground truth training data sets can lead to increased imprecision with which you generate for your final areas under the curve with your ROC maps. So any opportunity to automate the ground truth process is an opportunity for creating machine learning pipelines of greater sensitivity and specificity. So that led us to the idea of building a tool first and foremost to assist in the annotation of very, very large surface areas. This is very similar to Anant's group with the Rapid Annotator. I think that's what Anant's group is calling it. So these are complementary tools that basically leverage what a subject matter expert can do in picking a number of archetype or predicate representative images and then leveraging those over a much larger surface area or library of images of the same stain, so that an individual themselves, they don't have to manually paint all the regions of interest. And we'll go through a simple exercise showing that for even large areas of a whole slide image, if you were to try annotating an image at the pixel level would take inordinately large amounts of time. So a couple quick examples of some of the annotation tools that we've built over the years in the earlier versions of Viper, just to show its precision. This is an example where if you have, for example, micrometastases in a lymph node of breast cancer, you can either choose to use a library of pre-established spatial vectors, or if the morphology is unique, the feature classifiers we're using now will converge with as few as a small set of typical examples, which really diverges from some of the more typical use of convolutional neural networks, where you need large numbers of images to converge on a highly precise classifier. So this is kind of a whimsical extreme example where we're going to use a single classifier, which normally you wouldn't do this in production, but just to show you the sensitivity of the algorithm with a single classifier from this field of view, we'll apply it to the rest of the field of view and you'll see the level of sensitivity and specificity that's possible. So in this case, we're selecting a single nucleus and a single surrounding cytoplasm from the archetype. This is now being applied to this entire field of view in real time with our GPU cluster. And you can see that the ability to select all areas is very good. Maybe we missed a couple cell clusters here, but in general, to converge this quickly with a single classifier is not what one typically identifies. And then this, of course, allows for the identification of a matching montage of cases. Another time-consuming task, of course, is mitotic figure counting, counting mitotic figures for assisting in grading of various malignancy classes. In this case, you can again use a library of images, or if you have what you perceive as unique morphology here, so you can see a couple likely candidates, it's possible to again generate a classifier from a very small number of exemplars and then apply that to a large number of fields of view very quickly. And the goal here, so here in this case, I'm just selecting a single exemplar of a metaphase mitotic figure. The GPU cluster now will apply this to the entire field of view and grade the level of fit with the color of the heat map of the circle. So things that are very high are red, for example, less well fit are different colors. And then you see the pathologist is still very much in control in terms of up or down calling the various events. Some, of course, might be false positives. And you want an algorithm like this to be designed to be sensitive and not specific, so you can make sure you don't miss events. And then on multiple fields of view, again, applying the algorithm to identify multiple counts and then generate a running total across multiple fields of view, ultimately leading then to the total for that case. And you'll notice in this overall workflow, the advantage is you've shifted the workflow from being screening, which is time consuming, to directed review, which is much less fatiguing and provides for a more consistent practice. So now you've gone on multiple fields of view, as you can see, and you have your total count. And so this is just a simple example of how we envision that automation in the digital pathology workflow space will reduce fatiguing and repetitive tasks for pathologists to something which is both computationally tractable and from a workflow perspective, more reliable and actually more pleasant. So at the same time, elevating level of professional satisfaction. As an example, and this now we're sort of transitioning into what has been made possible very recently by the use of Viper. And this is sort of a preview of the actual interactive session I'll provide at the very end. This is the reference image. This is one of the kidney biopsies that we've been looking at in the Kidney Precision Medicine Project. For those of you who are renal pathologists, you'll recognize this as intermediate grade diabetic nephropathy, the typical glomerular sclerosis, glomerular injury pattern of diabetes. And this is, a subsampling of the native resolution of the scan, which is actually 40x. We can now apply the machine learning tools of Viper in mass to the entire surface area and classify every pixel at the pixel level to pre-made structures that are deemed to be either distinct or of diagnostic import, and then quantitate them. So this of course, at low magnification is perhaps suggestive of what's going on. But if I go at a native pixel zoom in of the actual resolution with which Viper assesses and computes images, every pixel has been subjected to a complex, not one, but set of machine learning pipelines and classified as to its best identity. In this particular case, we've done a k-means operation. Is it 100% perfect? No, not yet. We're actually still refining that. But if you were to take the number of pixels that were in that earlier field of view and do a simple thought experiment of 5 seconds by a subject matter expert to adjudicate each one of those pixels individually, that's 24.7 years, which of course can be done now in a matter of a few minutes. So this is the kind of computational scale that contemporary GPUs and computational clusters allow us to impart on the raw data, which is simply not possible before. Just as some quick examples of things we're doing within KPMP is annotating automatically all the key structures, for example, glomeruli, tubules, vascular structure, interstitial space. The ROC curves we obtain are really superb. As an additional example, we can subfractionate the interstitial regions, the finer interstitial regions, and then the more consolidated, as the second highlight shows, and then fractionate them separately and generate these pipelines with a relatively small number of training images, which differs to some extent from what typically might be required using conventional convolutional neural network approaches. So the use of Viper really is meant to reflect the fact that in many cases, pathology cohort sets of cases and images are not in the tens of thousands or thousands, you're in the tens or hundreds, and you still want to generate convergence to a very precise morphologic feature. So with a relatively small number of fields of view, for example, Dr. Laura Barasoni and I developed a spatial classifier to identify subnucleolar vacuolization in tubules, suggesting a relatively recent recovery from injury as one of the hypotheses. If you were to try generating this type of spatial filter with conventional means, in some cases it would be non-trivial. So this is just an example of the interactive nature of the Viper tool to very quickly stand up very precise morphologic descriptors. In tandem, one of the elegant things that we've been able to do within KPMP is educational tools as well. So for example, in this animation, we have a model nephron on the right and an example of histology on the left. And since this has been spatially mapped, as a student, for example, would wander their cursor over the image, the various areas of the nephron schematically light up as a simple medical school grade tool. And the type of educational tools that we envision building really will increase in complexity as we have more and more understanding of the individual function and biological multi-axial data from each location in histologies. We couple it to transcriptomics and proteomics, for example. And finally, I think one of the most fascinating aspects of having morphology now being linked with the various classes of omics data is the ability to crosswalk between morphology and the genomic space and vice versa. So in this early proof of concept in identifying particular genes of interest, since there's previously mapping of that data, the genes of interest now are correlated spatially to the areas of the histology that would be expressed. And again, this is very early stage data, but we're expecting within probably the next 6 months to a year, a trove of this from the various tissue interrogation sites that already now have very substantial spatially coupled information. So that this is now really an unprecedented window into morphology enabled by the other types of omics data. And of course, you can see right away that the inverse of this is possible, where a user could then select areas morphologically of interest, for example, selecting regions of a glomerulus, submitting them then to the same knowledge environment that's correlated morphologically the areas with gene expression, and then identifying the genes that are associated with that particular morphology. And again, tools like this are just beginning, but we know just by virtue of the fact that the data sets are available and coupled, it's a matter of execution to create the appropriate library of tools for both discovery and ultimately for clinical management, where the omics and the morphology are coupled together and serve the diagnostic functions of the pathologist hand in hand, as opposed to what we do now, which is typically look at individual immunostains as well as the morphology, and then piece the story together a little bit at a time without having all the parallel information in situ and available for parallel interrogation. So the high throughput technologies, by virtue of their parallelism, are really, I would predict, going to offer us a completely new way of looking at high dimensional data with an eye towards its morphologic underpinnings. But again, I would caution that approach to say very clearly that there still very much needs to be a physician and pathologist in the loop to oversee that adjudicative process, such that there can be appropriate biological context. That being said, I'm going to bring the Viper Studio tool into the screen. Anil, did this work? I'm making sure I'm not sharing my screen and not PowerPoint. Do you see the tool? Is it working?
Yeah, it's working. Yeah.
Great. Let me load an image, and I'm actually going to go through the
Well,
entire
I think we're still seeing the PowerPoint, aren't we?
No.
I'm
No,
hoping you're seeing
you're fine. You're
the
fine.
Viper Studio.
You can close the screen. Yep. Now, and if you can increase that, if you can make it bigger somehow.
I guess I could share the whole. Let me just share the application. The thing is, this application has multiple windows. So if I just share this application, I won't be able to share the other windows.
Okay,
So-
let's give it a shot.
Okay, so let's just go with what, because I'm going to zoom in to get some more resolution. So there's a high performance viewer built into Viper Studio. It loads multiple formats. Fortunately, I didn't have to write this. We largely base this on OpenSlide. We've added some extensions to it. But as a result, it allows us to focus on the computational discovery and not so much the whole slide viewer functions themselves. So in this particular field of view, just as an example, I have, let's see if I want to pick, this is actually a better field of view. So we have a tubule, we have part of a nodular sclerosis glomerulus. We can start generating some feature vectors. So again, the notion of Viper Studio is you make composite individual structural features which convert the image itself, which is really color pixels, into a metaspace where each physical pixel location is no longer a color, but represents the biological context. So you're creating a high dimensional space, which then can be processed by various traditional machine learning means. In this case, I'm going to be using as an example today, k-means, to make a spatial classifier at the pixel level, but is actually classifying biology, not just an RGB or an HSI color. So that being said, and then of the feature selectors, of course, you want to match their size to the length scale of the object being interrogated. So if I click on this, for example, I'm making, again, single constitutive vectors. I have a vector now, and you see it's not perfect. It doesn't have to be perfect. It just needs to be, in terms of this heat map space, close enough to the feature of interest that when running it through a principal component type analyzer, each spatial area becomes considered as distinct from all other spatial areas. So that's one feature. I'm not going to generate a huge number because obviously we have time limits of this presentation, but just enough to give you an idea of what Viper is capable of doing. So a feature vector of the nodular sclerosis. And you can see this is highlighting much brighter than everything else because, of course, it's the match for the nodular vector. I'll do another vector, for example, for the nuclei of the tubules. And we see that there's some different colors, a little bit of cyan as opposed to the green background. We'll do a basement membrane classifier. Get the one that's a little bit, that's pretty good. And we'll do maybe one more. Nuclei, for example. So the nuclei within the nodular sclerotic regions, that's pretty good. I probably have enough, oh, and then one, always white space as a final example. So the k-means can recognize background, so we can subtract that. It's always useful to have white space. At this point now, I can actually submit this, and we have a back-end machine learning engine that's running in the background, which basically takes the feature vectors. And I can choose now to divide this into as many high dimensional spaces as I'd like. So at this point, because these are hue, saturation, intensity triplets, and I have a total of 0 through 5 or 6 vectors, that's an 18-dimensional space, much more than the 3-dimensional space of just an RGB color, enough to run the algorithm. I'm actually just running this on my laptop. As a result of that, I'm not using the GPU cluster, but this just shows you how portable this application is. So those of you who choose to explore this application will be able to download this on your machines, and without exotic hardware, and certainly without a very large GPU cluster, carry out segmentation of not just individual fields of view, but when you're happy with your computational model, apply them to the entire field of view of a whole slide image. So while we're waiting for this model to complete, which will take approximately a minute or so, and we have definitely within time limits, right? Anil, I'm going to a 1, is that right?
Yeah, you are, yes.
Yeah, so I mean, I'm going to be done for questions in about 3 minutes. So, but just enough time to get the, I just wanted to show the output in detail. So, while the model's running, let me go to, and I showed this slide earlier. I'll just bring this image up. This is, I think, somewhere, the native information of somewhere around 35 by 75,000 pixels. So to expect that a human or a single subject matter expert would be willing to classify an image of this magnitude at the pixel level is probably unrealistic. I mean, and the reason the screen is still white is the image is so large, it's taking a while for the computer to bring it up. This is routine for Windows. Windows isn't really designed to bring up JPEGs that are so huge. So bear with me while we wait for Windows to catch up. I suspect also because I'm running Viper in the background, I'm consuming some of the CPU for that as well. Normally this would come up a little bit faster. One of the vagaries, of course, of live demonstration. Let's see if this comes up anytime quicker. Let me zoom out a little bit. So a note to self, when using Zoom and Viper and bringing up the Windows viewer, that is a very, very large lift for the operating system. Let's see if it comes back. So Viper is still going. Oh, it's back now. I just had to bring it off the screen. Okay. So at this point, I should be able to zoom in a bit more. There we go. It is working. So my point of just showing this is to give you a flavor for the level of spatial fidelity that a tool like Viper can provide in terms of classifying the thinner areas of basement membrane in red, the thicker areas, nodular sclerosis in the amber, the dark blue purple for the nuclei of the tubules, the cyan for the tubular cytoplasm. And in building this model with Dr. Barasoni, we also picked an additional lime green color to represent the obliterated areas of Bowman's capsule in the setting of diabetic nephropathy. What was elegant, and at first we didn't appreciate, so obviously you can see this is a diseased glomerulus with the obliterated Bowman's capsule. I'm not sure if I were looking at the PAS stain natively, I would have realized this, but this is a tangential cut, for example, of a glomerulus, and you can see we're catching some of the glomerular tuft as well. So the algorithm, because it's not looking at just color, it's now looking at the complex textures of each location convolved with color, as a high dimensional space, is providing a level of classification from information that's just in the PAS stain. So there's a lot more information in the PAS stain when you go to a machine learning based approach than what a human is going to directly appreciate. For example, these atrophic tubules have a very characteristic color pattern, which when subjected, for example, to a second pass of machine learning for k-means of the clustering of colors using Markov chains, you can identify all the structures of the kidney extremely tightly. And that's actually in the version of the program, which is downloadable. So at this point, we're still waiting for the k-means to finish, which should take perhaps another minute or two. ViperCrunch is running. Let's see where I can get that. ViperCrunch is still running. It should come back any minute. At this point, I think what would be the most effective use of time is I can take questions at this point, and then at the very end, I'll show how quickly one can make a computational model. So why don't I open up the floor to questions or comments, and then as time permits, I'll show how quickly one can generate a color-based model.
Thank you, Yul. That was great. So I've been monitoring the chat line, and if any of the participants have any questions for Yul, please add them to the chat line. I just wanted to, first of all, thank you. This was a great presentation. I think it helps for my colleagues in computer science to understand the emphasis that is needed for this workflow to occur, and the tool that you have presented obviously is going to require a lot of computing power. It's going to require a lot of infrastructure needs. So if you were, and I'm looking at the audience who's here so far from pathology and from computer science, if you had to ask our computer science colleagues who are experts in machine learning, they're experts in infrastructure, they're experts in cloud storage, everything else, what would your top 3 wish list would include in the wish list? So you're not asking this from pathologists, you're asking this from our colleagues in computer science.
Are you asking it to me or to the?
No, I'm asking this to you. If you are going to, if you wanted to implement this tool in one of the small community hospitals who is served by University of Michigan, and you needed your colleagues in computer science to help you, what would you ask them to do? What would the top 3 tasks would be for them?
So I would, obviously these algorithms are still very, very hungry for computational throughput. So what I would be very interested in is expertise to allow the conversion of the prototype algorithms, obviously to validate them. I mean, that's something that has to be done in partnership with other pathologists and biologists to make sure that they're identifying structures that are consistent. But in addition to that, effective ways of scaling things up and making use of standards. Because right now, if you look at, for example, what our group is doing, what Anand's group is doing, what Dr. Pinaki Sardar's group is doing, every group has their own sort of special flavor of GPU-based or high throughput cluster. And they're all a little bit different. And the lack of standards, both of the clusters themselves and of the data sets upon which we run on these clusters, creates essentially a friction in terms of discovery of being able to interoperate. And we've mentioned this at other retreat meetings of imaging scientists and biologists before, but I think it's still one of the paramount challenges for our field, which is having effective interoperable standards of the image formats, of the computational high throughput algorithm formats, and then of the platforms themselves, and then finally of the libraries for validating these pipelines so that groups can work together more effectively. And we really, I think, really need to leverage the idea of interoperability of programs, of training data sets, and of computational frameworks so that it's just easier to collaborate. Because right now, it's not completely an island unto himself, but we really do still have something of a silo, I won't say mentality, but a silo reality where groups find it difficult to interoperate, intercollaborate, exchange information. So I think interoperability necessarily should be one of our top priorities. And if I can just briefly interject, the model has come back. And this is where a subject matter expert could very easily generate a model and then apply it to anywhere on an image. These are the features in the high dimensional space that came back. And one can basically turn on features of interest. So I think that several of these are the nodular sclerosis, for example. So actually, these are the nuclei that came back. We'll identify maybe a couple more features. Actually, one of these might be the background, so we don't want that. Yep. So I've got basically now nuclei in one color and the Bowman's capsule in a different color, which we can dynamically reassign. And you can see very quickly now, a pathologist or subject matter expert can start assigning structures and colors. And so, for example, that's the cytoplasm of the tubule. We can make that aqua. And very quickly, a subject matter expert could have a complete pipeline of all the fields of interest. So let me make that also 11. So look, with just a very small number of clicks, and this is by no means complete, I've identified the tubule cytoplasm and the tubular nuclei, which then can be applied, for example, at any other field of view. Here's a nice field of view. We'll analyze this current field of view. The pipeline knows it has a completed model and is running this model now on this field of view and will generate in real time a classification. And once you're happy with your model and you lock it down, you can apply it to a whole site image or an entire field of view of a library of many whole site images. So let's see, it should come back any second.
So Yul, I have a couple of questions from the audience.
Sure.
While you're doing this. So
we just did it. Okay, yeah, happy to take more questions.
So there was a question from one of our attendees. Have you tried the tool on different data sets or cancers or other types of diseases?
Yes, it works. It works. So the tool, because it uses k-means and other types of like XGBoost, is completely agnostic to what it's looking at. It works on all stains, on all histologies, multiple magnifications, and it even works on MR, CT data. So it works on radiographic data. Because again, the algorithm itself fundamentally is looking at color and texture. It doesn't even know it's looking at biology. It's simply convolving the local textural milieu with the local colors and generates a higher dimensional descriptor other than color. For it's textural convolved with color. So yes, it works on cancer very well, actually.
Excellent. So there's one more question. Can you give us an idea of the system on which these models have been run? How many GPUs? What are the sizes of the images and the ML models?
So it runs on a machine that's simple as a Windows 10 machine with 4 cores, 8 threads. You can see it's taking advantage of the multi-thread. It will run on larger machines, so up to the current CUDA library. But it doesn't, so the thing it needs more than anything else is just memory. The minimum that it will run on is an 8 gig system, which is a pretty routine system at this point in time. And architecturally, it's designed to scale up linearly with both CPU and GPU performance. So we've designed this specifically with the intent that it could operate in an entry-level workstation and then go up from there.
Okay, so there are some more questions on the chat line. If you can respond to them while I'm going to introduce our next speaker, because we want to keep track of, we want to stay on time.
Yeah, very happy to.
So thank you so much for the presentation and sharing this great tool with our colleagues. So I'm looking forward to, you participating and exchanging these tool sets. And you have some other questions that are just coming up the line. So please try to address them as soon as you have some time.
Very good.
Thank you.
Thank you very much, Anil. I'll answer questions right now.
All right, thank you. So with this, I'm going to